Good day, everyone, and you're welcome back to our classes this afternoon. I hope we've been enjoying the classes as we've been putting out the different videos. Now, today we are going to discuss an interesting topic. We're going to discuss about viruses. But before we start, I just want to say or give a shout out to a senior colleague, Professor McDonald Igodaru, who has been following these videos and offering suggestions and corrections along the way. Sir, I do appreciate your help and I'm grateful for your comments and your recommendations. Now, today we are discussing viruses. What are viruses? Viruses are what we refer to as acellular entities. They are acellular entities because they are entities that form a bridge, a gap between living things and non-living things, If you look through the videos that we have been putting out, you see that when we did the five kingdom method of classification by Robert Whittaker, we did not have any place where we put viruses because viruses have always been a problem for scientists. Where to put them and where to fit them in has always been an issue because many of the characteristics they have are borderline between living and non-living organisms, making it difficult to place them in a particular category. But today we'll look at viruses in totality and just try and discuss a little bit of their features and characteristics that make them unique and what viruses are all about. First, they are microscopic in nature, and so they cannot be seen by the naked eyes. Now, they are so minute in nature that they have to be seen, they can only be seen by microscopes, and not just ordinary microscopes. They are seen by very large and powerful microscopes, an example being the electron microscope. The electron microscope has the ability to really magnify objects. When we get to discussing microscopy, we'll see that in microscopy, there are two issues. There's magnification and there's resolution. So the electron microscope is able to really magnify an object, that is, make the object look really much, much bigger, many times than it actually is. And resolution has to do with the issue of if two objects are side by side, are you able to really, differentiate them when you look into the microscope to know that this object A is different from object B, or this organelle or this feature are different. So the electron microscopes are used to view viruses, and viruses are microscopic in nature. Now, viruses, when they exist outside the living cell, they take up a crystalline nature and they are not able to function. They don't do much in the environment except they find their way into a living cell. Now, let's look through some of the characteristics of viruses and why viruses are important. They are microscopic in nature. Now, secondly, viruses have genetic materials. They have either a DNA or an RNA as their genetic material. We know that the DNA is double-stranded and the RNA is single-stranded. So you could have DNA viruses or you could have RNA viruses. This is a very important feature of viruses. Now, viruses also have a protein coat. The protein coat is called a capsid and is very, very important. Viruses have this protein coat, which is called a capsid. Now, the capsid gives the virus their shape. We have many different shapes that viruses could take. There are viruses that have helical shape, so it's shaped like the DNA molecule. There are viruses that have what we call icosahedral shape. Now, the icosahedral shape is unique because it's a shape where you have 20 faces of an equilateral triangle, 20 equilateral triangles all around the structure of the virus. And there are viruses that have such a shape. So we also have viruses with what we call a complex shape. The shape is difficult to describe. So, and it is the capsid, the protein coat of the virus, that gives the viruses these different shapes that they have. Now, the capsid, the protein coat, also is made up of little subunits of proteins that we call capsomeres. Capsomeres are the little subunits of proteins that we bring together to form the capsid, which is the protein coat for the virus. Now, viruses do not go through a lot of all these metabolic activities that other organisms go through. But viruses are not able to undergo binary fission. They are not able to be metabolically active like other organisms. They don't respire, they don't feed, they don't excrete. Now, some viruses have what we call an envelope. So sometimes you could classify viruses on the basis of the presence or absence of the envelope. So we say some viruses are enveloped viruses while others are non-enveloped viruses. Now, what is the envelope? The envelope is a liquid substance, a liquid material that surrounds the virus. It is all over the protein coat for a virus that has an envelope. So the thing is that what makes it important is when you look at medical significance of such a thing, you realize that for the virus that has an envelope, such a virus is more resilient in the environment, is able to survive longer, it's able to cause more virulent diseases because of the presence of the envelope. So you have enveloped viruses and you have non-enveloped viruses. Now, viruses can also be classified in quite a number of ways. One of the ways is the kind of organism that the virus affects. We have what we call animal viruses and plant viruses. Animal viruses are viruses that can be found, just as the name implies, majorly in animals. So when you think of a very simple and common example, the rabies virus, we see that the rabies virus can be found in dogs, and it could, if those dogs bite humans, such viruses can be transmitted to humans to cause rabies disease. So this is an animal virus. Meanwhile, you can also have what we call a plant virus. An example being your cassava mosaic virus that causes cassava mosaic disease in cassava. So such a virus inhabits, and it can be found in plants and not in animals. That's one way of classifying viruses. And earlier, like I said, you could also classify viruses on the basis of the kind of genetic material which they carry. If the virus is carrying a DNA molecule, we say it's a DNA virus. If it's carrying an RNA molecule like you have in your retroviruses, like your HIV virus, we say it's an RNA virus. So this is very important. Now, viruses have no organelle. Remember, we discussed what an organelle is. These are membrane-bound structures that can be found inside cells, and they carry out particular functions in cells. Viruses do not possess organelles of any kind. And like I earlier said, if they are outside a living cell, the viruses take on a crystalline form and they are inactive. They are unable to do anything until they find their way into another living cell. And this is very important. Now, it's also important to know that there's what we call a viral life cycle. That is, the viruses have a life cycle which they follow. Now, what's this life cycle? The first one there is attachment, entry, replication, assembly, and release. Now, in attachment, viruses have what we call glycoproteins. They have spikes on the capsid, and these spikes, they use them to attach to the living cells which they want to find their way into. Now, that process is called attachment. When they attach to these living cells, now the virus is able to inject its genetic material, let's say it's RNA or it's DNA, it injects it into the living cell that it wants to colonize. So that process is called entry. So there's attachment where it uses its glycoproteins, its spikes to attach to the cell surface proteins on the living cells that it wants to attach itself to. And when it does that, that is attachment. Then when it injects its genetic material into the living cell, that is entry. Now, after entry, you now have replication. What do you have happening in replication? This viral DNA or viral RNA that has found its way into the living cells now begins to look at all the machinery, all the genetic machinery that living cell possesses. It begins to take up those genetic machinery and begins to use them to make replica of itself. So the DNA virus or the RNA virus, when that DNA or RNA comes into the living cell, when it gets into the living cell that the virus wants to colonize, it now takes up all the genetic material that the cell has, that it is using for itself. The living cell is using for its own metabolic activities. It takes up those genetic materials and forms replica of itself. So you now have a lot of DNA, viral DNA or viral RNA through replication. After replication, there is also assembly. That's within the living cell. You now have there's the assembly. So what you now have is that protein coats start to form around that the new viral, DNAs or RNAs, which are still within the living cell. It also takes up protein from the living cell and forms these capsids. And after that, there's the release. Once it has made use of all the machinery, all the resources of the living cell, of course, cannot sustain life anymore due to this process. And then it releases, or there's a release of viral particles, and all these viruses then leave this living cell. And of course, the living cell has been destroyed by this process. So there's attachment, there's entry, there's replication, there's assembly, and there's release. Now, I'll end this class by discussing that viruses have medical importance and medical significance, because a lot of viruses cause a lot of diseases, both in man and in animals. And even viruses also have application in agriculture, because there are a lot of viral diseases that occur in plants and even in animals. So all these things are important. The life cycle is important, and this is what viruses do. I hope we have understood a little bit of viruses and their structure and what they do. Thank you for listening to this class.